In this video, we're going to have a look at capillary beds, how they work and their structure. So let's get to it. So capillary beds really are this network of capillaries that connect arterioles and venules. And we can think of this as a microcirculation, flow of blood through the bed from the arteriole to the venule. We also have these terminal arterioles. They branch off arterioles and further branch into somewhere between 10 to 20 capillaries. And of course, the capillaries are where we're going to do the exchange. And in total, this forms a capillary bed. And we'll have a look at a diagram very shortly. Now, what's happening here? Not surprisingly, we're going to exchange gases, nutrients, wastes from the surrounding tissue, and this is what takes place at capillaries. Now, capillaries then drain into the post-capillary venule, and that's where the blood will end up after going through capillary beds. Now, not all your capillary beds are open at once. If they were, the container size would be too big for the amount of blood in your body, pressure would drop. So we control where the blood goes. So if you don't need a whole lot of blood, the capillary beds aren't really open in that area. If you need blood in that tissue, well, then we open the capillary bed. So it's a very dynamic process. So we have flow through the capillary beds, and it's controlled by diameter of the terminal arteriole and the upstream arteriole. So by changing the diameter of the vessels leading into capillary beds, this helps to control whether the capillary bed gets blood, basically. Now, there are local chemicals and conditions that have a role on terms of this blood flow. So local chemical conditions and the arterial vasomotor nerves, okay, they regulate the amount of blood entering the capillary bed. So we really need the arterial and terminal arteriole dilated when blood's needed into the capillary bed, and then these are constricted when we do not need blood to go in there. So let's have a look. So here we have an arteriole, and from an arteriole, coming off an arteriole, we have the terminal arteriole that heads in the capillary bed. Then we can see the capillary bed. It bifurcates all the capillaries here. And then blood is collected at the far end, and we have the post-capillary venule, and blood gets returned. Now, as I said, capillary beds are not open all the time. Sometimes they are closed. And a lot of this is controlled by the arterioles that are leading into the capillary bed. So if it's fully dilated, resistance is low in that vessel, more blood will head here. If it's vasoconstricted, then less blood is going to head here, and functionally the capillary bed is closed down. Now, that being said, let's look at some more aspects of it. So capillaries found in serous membranes of the intestines have two additional features that form a special arrangement, and one's called the vascular shunt, and this is a channel that directly connects the arteriole with the venule, and it bypasses the true capillaries. We're going to have a look at a diagram in just a few seconds. It consists of two parts, the meta-arteriole and the through-fare channel. So these are two components of what we're going to consider the vascular shunt. Now, there are these precapillary sphincters, and these are awesome. They're these little rings of smooth muscle that surround each capillary as it branches off this thing that we're going to see in a second called the meta-arteriole. And this is going to act as a valve that's going to regulate blood flow in there. Now, this is controlled by local chemical conditions and is not innervated. So let's have a look at this. Now, remember, this is not happening in every capillary bed. This is a special group, like the mesenteric capillary beds. And you can see I do have the arteriole, I do have the arterial heading in here, but in this case, what's different is there's a channel that runs through to connect both the arteriole and the venule. And then how do I control the flow through the capillary bed in this very special route? I have these precapillary sphincters. So if the sphincters are closed, then of course blood will not transit through here. If the sphincters are open, then blood can pass through. And that's what's going to control the capillary bed itself. All right, so let's talk a bit about what is happening here in the capillary bed. Now, let's start with this diagram. So what I've tried to draw here is basically, let's walk through it, is that we have the arterial leading into the capillary, and then the capillaries, if they're open and blood's flowing through them, blood will go back into the venous part, right? So one thing to consider is the water movement, because where water goes, other things get dragged with it. Now, there's some other things that happen within capillary exchange that we will have a look at, but I just want to introduce this concept of the water movement. Now, what is the force? Now, we have learned that capillaries do have little openings here, little spaces, and these are our normal continuous capillaries, so water can leave. So what is the force that pushes water out at the arterial end of the capillary bed? It's BHP. Now, remove the H, and that looks familiar. Looks like BP, blood pressure. This stands for blood hydrostatic pressure. So that's the water pressure that's going to force some water out of the blood. And of course, we want water to go into the tissue, to hydrate it, to bring nutrients with it, etc. Now, something else we should consider is I drew these little green squiggly things. These are to represent the proteins such as albumin. Albumin's the dominant one, made by the things like the liver. And these proteins are too big to leave, so they don't leave. So as I'm losing water, I'm not losing albumin, but because I've lost water and I've kept the albumin, functionally I have a higher concentration of the protein albumin at the end of the capillary that's closer to the venule. And what this does is the water that left here, 85% of that water will be reabsorbed at the venous end of the capillary. Now, what's the force that's bringing the water back in here? Well, remember, we have this increased concentration of albumin. Remember, I didn't make more albumin in this short distance. It's just that I lost water. So functionally, I have an increased concentration of albumin. And technically, because this albumin is in a watery solution, it falls under the category of a colloid. And this is an example of colloid osmotic pressure. So the mere fact that we have these proteins largely dominated by albumin will pull the water back in. Now, we'll pull the water back in. Now, you're probably thinking, okay, well, what did I accomplish? Well, by having the water go out and in, I'm bringing some of the nutrients with it. I'm bringing waste back in here, and I'm hydrating the tissue. Now, you notice I'm only collecting 85% of the water at this end, which means there's potentially a buildup of 15% of water that was not recaptured down here. Now, luckily, this is where lymphatics come in, and lymphatics are another chapter, but lymphatics are very much like a vein, and lymphatics will collect the extra fluid and run it through a series of lymph nodes before returning that fluid to the blood. So this water isn't lost to the blood, it just has to take an indirect route, a longer route to get back into the blood. So that's a little bit about the water movement within capillary beds. I just want to introduce that concept. So we're going to end the video here, and then we're going to come back and start to look at veins themselves. We'll see you in that video.